computer so I can have control of it. Okay. All right, take it away, Kristen.
Okay. Hi, everyone. I'm Kristen Garcia. I'm a third-year PhD student in the bioengineering department, and like Kelly said, it's my second year on the training grant. I'm currently working in Dr. Valdez-Hasso's lab, and I'm interested in using a three-dimensional biventricular model of the heart to answer questions around sex-dependent differences in pulmonary arterial hypertension. So an outline for my presentation, I'm going to start by introducing pulmonary arterial hypertension and explaining why it's important. And then I'll go over the experimental protocols I'm currently working with, which relates to my current work and goals. And then I'll end with my future goals for this quarter. So pulmonary arterial hypertension is a life-threatening progressive disease that is characterized by pulmonary vascular remodeling and is represented by the narrowing of the blood vessels in the lungs. So the pulmonary arteries are the blood vessels that take blood from the right side of the heart throughout the lungs. And if you look at this picture right here, you can see on the left-hand side, it's the pulmonary arteries of the normal heart are right here, and you can see they're wide and blood is easily being able to go through. But then when a person has pulmonary arterial hypertension, that's when these pulmonary arteries constrict. So it's harder for blood to come through, and then the right ventricle has to adapt and work harder to get the same amount of blood through to the heart. It's described by a mean pulmonary arterial pressure of above 20 millimeters of mercury. And so in the United States, there are about 500 to 1,000 new cases of PAH each year. And this disease is most likely to affect women between the ages of 30 and 60 years old. So this leads to like an interesting paradox because women are 4 times more likely than males to get the disease. However, they're also more likely to survive the disease than men that get it. So this brings questions around estrogen, which is the main female sex hormone, and its role in PAH. Past research has shown contradicting results. So some past literature has said that estrogen has a protective effect over the heart, and that's why the women survive better. But then others have shown estrogen to have a disease promoting effect, and that's what causes females to have more likelihood of PAH. So to study PAH in our lab, we use a rat model, and we use a known Sugen hypoxia protocol that has been is widely known and used to induce the pulmonary arterial hypertension. So pretty much we induce Sugen 5416 into the rat. And so this is a vascular endothelial growth factor receptor inhibitor. And so after we inject it, this is followed by 3 weeks in hypoxia, which is 10% oxygen levels. And then the rats return to normoxia, which is just normal air, and the disease continues to develop. And so we work with 3 different groups. We have a group of male rats, a group of female rats, and then a group of overrectomized rats. So the overrectomized rats have their ovaries removed, and this is to represent women with menopause, like going through menopause or past menopause. And so for each of these groups, we have control and then the treated group. And so at different stages along the progression of the disease, we have different cardiac imaging done on the hearts of the rats. So we have cardiac MRI and DTI imaging done to look at the geometry of the heart and the fiber orientation throughout the heart. And then we also collect hemodynamic data during surgery. So we use pressure volume catheters during surgeries to measure pressure volume loops, which is the gold standard for heart function, is to see the cardiac function. After these surgeries, we remove all the tissue. So these are terminal surgeries. And then the right ventricle is run through a planar biaxial testing protocol. And so that is when we look for stress strain measurements and determine the stiffness of the tissue. And then we also run the tissues through different histological analysis and assays to determine collagen content, ECM composition, and different like protein quantifications. And then the whole goal is to put all of this data together in a computational model that can help us predict the outcome of the disease and why it's different between males and females. So this slide shows up kind of a pipeline of what I'm currently working with. So we have cardiac MRI done here at UCSD. So it's through the Molecular Imaging Center at Stanford. And so we get the cardiac MRI done, and then this is in the form of a DICOM file. And so we input this DICOM file into the program called ITK Snap. And in ITK Snap, I am manually segmenting the left ventricle from the right ventricle and the epicardium. And so after this it's manually segmented, I move these 3 files, which are STL files, to Autodesk Mesh Mixer, which is just another software in which I can smooth out the ventricles and make them look realistic. Because since I am doing manual segmentation, it is a little rough around the edges when I do it. But then after I get the smooth ventricles, I move to Gmesh, which is a free open source software that is a generator for 3D finite element meshes. And this is what creates the 3D mesh of the heart. And so I'll go through each of these steps. So we currently have scanned 3 different groups of animals, so male, female, and ovarectomized animals. And we have them scanned for cardiac MRI at 3 different time points. So when they're control, and then 4 weeks after PAH was induced, and then again 4 more weeks, so a total of 8 weeks after PAH induction. And so the whole goal of this is to be able to create 3 different meshes for each animal to see the difference in what the heart looks like and how it functions over the progression of the disease. And so these cardiac MRI images were done in the short axis slices, so from the apex of the heart at the bottom all the way up to the top and the base of the heart. So there's about 8 slices for each heart due to the size. And so these scans were also ECG gated, so we were able to see when the end systolic and end diastolic volumes when they were occurring. And so this is important because the end systolic volume is the smallest volume when the chamber's at the smallest volume, and the end diastolic is when it's the largest volume. And so we're able to calculate all the volumes for all the chambers using this cardiac magnetic resonance disk summation method. So pretty much what this method does is I just take the area of each slice and then I add up all the areas and multiply by the slice distance to get the total volume of that chamber. And also with these cardiac MRI images, we're able to look at wall thicknesses for like the left and right ventricle and see how that changes over the progression of the disease, as well as calculate stroke volume and ejection fraction and compare those within the one animal and across different animal groups. So this is an example of one of the yes.
Are you doing questions at the end or no?
At the end. Okay, so this is an example of one cardiac MRI image that we got, and so I labeled the RV and the LV, and you can see they're moving on the right hand side. And so the left hand picture is the end systolic slice. So in this slice, it's when the heart is empty, the chambers are empty because it's fully contracted and it's the smallest volume. And then for the end diastolic slice, these are the largest volumes of the heart because the ventricles are fully relaxed and filling with blood. So then we can just look at the video again, and so you can see the heart is beginning to fill with blood, so that's why the chambers are getting bigger, and then the heart is ejecting the blood and it's moving throughout the rest of the body. So we input that image into ITK Snap, and this is where I do the manual segmentation. So in order to do the manual segmentation, I just outline the left ventricle, right ventricle, and the epicardium. So you can kind of see right here that this is me outlining the left ventricle, and then you just fill it in right here, and you do the same with the RV and the epicardium, and then it creates this 3D image below, or segmentation below. And so we could have done, so this program does have automatic segmentation, but my images were not to the best image quality resolution, so it wasn't working as efficiently as me just going through and manually segmenting because there's only 8 slices. So next we put those 3 slices, so the LV, RV, and epicardium into this Autodesk Mesh Mixer. And so this is a program that pretty much just smooths the ventricles. So you can see that going from here where they're very like rigid and rough, and it's just not nice and smooth, I just use some brushes that the program has, and you're able to smooth it to the shape that you want it to be in. And then I take those 3 smoothed RV, LV, and epicardium and put it into Gmesh. So this is just generating the 3D mesh. So this is the 3D mesh of it. And you can see the mesh on the left and the mesh on the right are different. They have different numbers of elements. And so this is just something I can control depending on what finite element analysis I want to do. So you can control the amount of elements within the mesh. And so from here, this is where I want to interpolate the fiber directions onto this heart so we can see how the fibers look within this mesh. And so that's where diffusion tensor imaging comes in, also called DTI imaging. So the whole goal of this was to determine the geometry and the fiber orientation in these hearts. So we got this done at the University of Utah Small Imaging Corps. And so we sent 2 male control rats and 1 female control rats to University of Utah, and they were able to scan them and do some post-process analysis for us. We did harvest the hearts at UCSD, so in our lab. So we harvested the hearts and then fixed them with 4% paraformaldehyde in PBS, and then sent them to the University of Utah in just 1x PBS so that they didn't dehydrate. And so on the screen is one of the original post-processing analysis that this University of Utah sent me. And so you can pretty much see how the fiber directions change from the epicardium to the endocardium layers of the heart, and it does not stay the same throughout the heart. And it just shows the short axis and long axis slice. Because something to note is for the DTI, it was done in long axis slices, not short axis like in the cardiac MRI. So in this middle picture, you can kind of see the difference. So the cardiac MRI was done like A and C, whereas the diffusion tensor imaging was done in the long axis slice, as in B and D. And so the left-hand picture is one from the middle of the heart. So you can see both the right atrium, left atrium, RV, which is right here, and then the LV. And then the different colors, how it goes from black to white and gray, is a measure of the fractional anisotropy. So it's like the degree of anisotropy within that given voxel. And so the purpose of this is like a main measurement in all DTI imaging. And so for us, it can help describe like the underlying myocyte organization. And so an FA of 0 means there's isotropic diffusion, where FA of 1 means there's no diffusion. And so something to note is that for most fibrous tissues, the FA is normally between 0.3 and 0.6. And all of these values were just scaled as image intensity. So that's how we get the black to gray scale. And then this image is just me slicing through the z-axis, so it's just the thickness of the heart, so like anterior to posterior, and it's just showing how the fractional anisotropy changes throughout the location of the heart. So after the imaging is done, terminal surgeries are performed. So this is when we get the hemodynamic data. So we insert pressure volume catheters into the right ventricle of the heart, and then it allows for us to see pressure volume loops. So in B, you can see 3 pressure volume loops. So these are all for 3 different animals. So each loop corresponds to a different overoptimized control animal. And pretty much are just showing the shape of what a PV loop would look like. And so these are all over 1 heartbeat. And after each steady state PV loop, so each of these is 1 steady state PV loop. So after each of them, we do an occlusion during the surgery. So that is the gold standard for measuring like load independent contractility. So when we do the occlusion, it helps us learn about the contractility of the tissue. We also use other catheters like the dual pressure catheter to measure the pressures in the pulmonary artery and right ventricle at the same time. And this allows for us to get the mean pulmonary arterial pressure and see what it is. And we also have a flow probe that measures the blood flow in the aorta and pulmonary arteries. All these PV loops can help us like learn information about how the heart is functioning. So it can tell us stuff like the stroke volume, cardiac output, ejection fraction, and systolic pressure volume relationship, and diastolic pressure volume relationship. And so we can use these to compare between the different groups and see how if one animal group all has like relatively similar stroke volumes compared to other groups. And then after these surgeries are done, we take out the right ventricle of the heart and we cut out a piece from that ventricle, and it's normally the same like square piece every time, like that's what they try to do, and that's run through the planar biaxial testing protocol. So that's like when we stretch it in 2 directions and get the stress strain measurements, which can help tell us about the stiffness of the heart and to see if RV tissues get stiffer over the progression of the disease or just between different animal groups. So I'm also interested in looking at the estrous cycle more and the estrogen levels within the heart. So the estrous cycle in rats is similar to the menstrual cycle in humans. The timing is way different, but it's kind of the same concepts. And so throughout the different stages of the estrous cycle, like in this graph, you can see the stages up top, and then you can see the levels of hormones change throughout the stages. And so this is crucial for us because if we're saying that estrogen is making the heart function better, and we do the surgery at a time when estrogen is low and we get the different results, we're not actually seeing how estrogen is affecting our results. So it is important to understand and for us to know what stage of the estrous cycle the rat is in, so we can correlate that to the levels of hormones and what we expect to see in our conclusions. So we don't actually know right now what stage of the estrous cycle the rats are in when we do the surgeries, but we're trying different ways to see if we can tell this. So one way is to look at how the uterus weight corresponds to the estrous cycle. So it has been found that the estrous cycle changes the uterus weight, as you can see up here. So in diestrus, it's much longer and smaller, whereas like in proestrus, it seems like it's more inflated. So there have been papers that have shown that there's different weights that go along with different stages. So currently, right now, after every female and ovarectomy surgery, I remove the ovaries and the uterus, and then we just pat them dry and weigh them. And so this is with hopes of this having some kind of inclination of what stage they're in. And so these are kind of the graphs from what we see so far. So the left graph is all the uterus weights for each group, so like the averages, and the sample size is on the bottom. So you can see that for female control, we only have 2, so it's not really a good representation yet. But what we can tell is that females overall do have a higher uterus weight than the overrectomized uteri. So that's something that we do know, but we cannot conclude if the Sugen hypoxia protocol has any kind of effect yet. So we're gonna hope to, like, I'll still be collecting them after every surgery to get more samples, and then we'll hope we can see like a bigger difference and can conclude if like Sugen hypoxia has an effect. And then the right graph is the same graph. It's just all of the uteri weights are normalized to the body weight of that animal. So for future steps, I'm currently trying to interpolate all the fiber directions onto the mesh of the heart. And so I'm starting with just interpolating it onto this specific square that's being biaxed. So if you see in this top green, so this is just a model of the DTI heart that would like to be imaged. And so the 4 dots represent the corners of the biax piece that was biaxed. And so I'm hoping to just interpolate all the fibers to this small biax piece, which would then hopes it would make it easier to interpolate it to the entire heart. I'm also interested in looking at isoflurane rates and how it affects males and females differently. So during our surgeries, we anesthetize with isoflurane, and we've noticed that during some surgeries, we have to change the level a lot more than during other surgeries. And so we're starting to record this. So the standard, what we normally do is 2.5% isoflurane, but then we've noticed that some rats just continue to wake up during surgery, so we have to increase the isoflurane level. And then others rats, the heart rate will drop really low, so we have to decrease the isoflurane level. So this is just starting to get an idea of what the difference is between these male and females and overrectomized females. And so you can see like this tall gray line. So this is the female control animal. So over 54% of the time, we can leave the isoflurane rate at 2.5% for this group. So it doesn't really have to change at all. But then for like OVX control, so overrectomized control animals, 50% of the time we have to change the ISO level at least twice. So we're trying to see what else is different between males and females and overrectomized animals that is changing the way they react to just our anesthesia protocol. My last future step is we're going to be starting to put temperature sensors into the overrectomized and female rats. And so we're hoping that with these temperature sensors, they continuously monitor the temperature of the animal. So this will give us more insight into what stage of the estrous cycle the rat is in. So if we continuously monitor their temperature and can tell when their estrous cycle is, then we can control when we do our surgeries and make sure all the surgeries are either at the same stage or we purposely do them on specific days. And with that, just my end goal is to look at the differences between males and females as a whole to see how and why they are PAH affects them differently. And that's it. Yes.
What's the proposed mechanism for why estrogen might help with PAH?
Not off the top of my head.
In like humans, do you see a difference between women who are in menopause and get PAH, or is that irrelevant because most women get it about the same time?
They're saying that I think menopause is more similar to the male, so that's why we have like the overactive rats are more similar to the male rats in comparison to, so they're not getting it as often.
So there's not a number of heart diseases and vascular diseases. There's studies suggesting that estrogen can be predictive in pro-fibrotic remodeling, so that women who are premenopausal often have less severe pro-fibrotic pathologies. And that also seems to be something that can happen in some of these animal models of heart disease and cardiac remodeling.
But does the reverse hold with testosterone? Because I believe testosterone is also pretty anti-inflammatory for.
Yeah, some studies look at E to T ratios and others look just at E2. So I don't know that anyone's shown a protective effect of testosterone. Kristen, if you go back to your finite element mesh. If I look at these, you see these sort of regions where it looks it's hard to tell for sure, but where it looks a bit like the elements could be kind of distorted, like very elongated and stuff. And I just want to let you know that the Holst lab at UCSD, Michael Holst, his lab developed a tool a number of years ago that they've refined called GAMA, which is generalized adaptive measure. And one of the things it does is it looks at measures like this and it cleans them up. And the reason they do it is specifically to improve the numerical stability and convergence of finite element solutions. So they're not just doing it to make it look prettier, but because these distortions often give rise to numerical problems in the finite element solutions. So the GAMA tool is one you might want to look into.
Thank you.
You said you injected a growth factor to the mice. What is the what does it do?
So we're putting it in so it prevents the vascular endothelial growth factor receptor inhibitor. So it prevents them from growing, and so it makes them constricted, and then the hypoxia keeps them constricted, and then when they leave, the sugen is what also doesn't let them just go back to the normal size.
So hypoxia is a vasculogenic stimulus. But if you combine hypoxia with a VEGF inhibitor, it's not a growth factor they put in, it's a growth factor inhibitor, then you get sort of maladaptive vascular remodeling and these plexiform lesions that are commonly seen in human PAH, and that's what makes this rodent model the best model, because it has this sort of characteristic rarefication of the vessels and plexiform lesions. So
So is it to induce the pH into the rats, or is it to
Yeah.
just stabilize them in that state?
Induce it.
Yeah, hypoxia alone wouldn't induce it. Hypoxia alone would make the pressure go up, but it wouldn't induce the pAH pathological phenotype, the vascular rarefication. In other words, they would just go back to normal if it wasn't for the growth factor. Inhibitor.
With respect to this model, it's got me thinking about the genetic component as well. And something that I think is really fascinating is that the difference between humans and chimpanzees, our closest relative, there's about a 5% genetic difference. But when you look at men versus women, because we have that Y chromosome in males, right? We have a huge chunk of DNA that we just don't have expressed in males. So between men and women, it's about a 5% difference as well. Has there been any efforts to maybe in the future direction look at kind of something that may put men more at risk for this?
Can you repeat your question? Wait, so you're saying, so we normally look at we're looking at what's causing it in females, but you're saying to put them at risk of not surviving it as well.
Right, yeah, like beyond estrogen, would we look at other?
Yeah, I'm trying to look at everything as a whole because estrogen people are showing both ways. So I think there's more than just estrogen that's causing PAH more in females and having them survive more. So yeah, I'm interested in getting other views of just the human body as a whole, because I don't think it's just estrogen is the reason.
Anybody else? I'd briefly like to introduce Dr. McCullough, who is the program director. Raise your hand, stand up, of the Interfaces program. And so if anybody has any questions, Dr. McCullough, I'd be happy to help you. Alex, would you raise your hand? Alex is one of the new trainees, as is Justin. Justin, raise your hand. You know Joseph. Here's Daniel back here. Who am I missing? Am I missing any of Bianca? Where are you? There she is. Okay. And of course, you know Kristen. So those are some of the new trainees this year, and we're excited to have them here. And Dr. McCullough can tell you, as he's been telling you, more about the actual science of this. I don't have any idea about that. Anybody got any more questions?
Okay, so welcome everyone, and the purpose of these meetings, and really the purpose of the whole program, is to promote interdisciplinary interactions. You're all learning in a very interdisciplinary environment and going into a future that will be even more interdisciplinary. And UCSD has always been really good at this. But in the early days, the way we were good at it is that we started one of the first neuroscience departments and one of the first bioengineering departments and one of the first chemistry and biochemistry departments. So one of the first nanoengineering departments. And so, but can't keep on doing that forever, right? We can't have a nanobiochemistry, physics, medicine department, right? So at some point, the traditional structures are the structure that we can rely on to promote interdisciplinary collaboration. And so sort of in the campus world, I direct what's called an organized research unit, the Institute of Engineering and Medicine. And the ORUs, like the Institute of Engineering and Medicine or the Institute for Neural Computation or many of the other institutes, they all have this feature on campus that they promote interdisciplinary research and support it and facilitate it. And in the graduate level, we do have some interdisciplinary graduate programs, like biomedical sciences, for example. But then they themselves sort of are organized around a discipline. And so to promote even greater level of interdisciplinary interaction, we have these PhD specializations. And so our PhD specialization, which by now probably all know, is in multi-scale biology. And then the question is, what do we mean by multi-scale biology? And, this grew out of discussions with people in the medical school, particularly pharmacology biology, Professor Susan Taylor, and chemistry and biochemistry, particularly Dr. Andy McCammon, and neuroscience, particularly Dr. Mark Ellisman, and biological sciences in the Salk Institute, Terry Spanowski, and others like Romeo Amaro. And so many of us worked together on different projects already, imaging projects and computational projects, and we kind of had come together on this multi-scale theme, multi-scale imaging, multi-scale measurement, multi-scale analysis. And so that's part of the reason that we came up with this theme. Then the question was, well, how do we teach multi-scale biology? And we really decided that, you know, one of the sort of key enablers of multi-scale biology was technologies at different scales, right? The reason that Abby can think about making a model of the heart that starts with atomic resolution models of actin-myosin and ends with pressure volume curves like Kristen was showing. The reason she can even contemplate that is because we have the tools to do it. You know, Dr. McCammon with Tarpless and McCammon, and Mark Tarpless, the Nobel laureate, developed molecular dynamics in 1977. And then the McCammon lab developed new tools for accelerated molecular dynamics, Brownian dynamics, and then the Amaro lab came along and developed new techniques to allow us to scale up from the atomistic scale to, you know, in the case of the Amaro lab, now they can make models of an entire, you know, viral particles. Meanwhile, in my lab, we started out making models of the heart, and then we realized we could make more detailed models of the tissue, and then we made more detailed models of the cell, and then we started to bring in models of molecular events, dynamics, and start to put all of them together. And so bit by bit, we realized we had the computational tools to span scales of biological organization from molecule to organism. And actually, there are people who do population biology too, mathematically. So, but we also realized that this type of work, it's enabled by measurements. It's enabled by measurements in structural biology, enabled by measurements in techniques like electron microscopy and electron tomography, you know, super resolution light microscopy, live cell imaging, tissue engineering, in vivo imaging, the MRI, and as well as modeling. And so we constructed our program to, you know, give you access to hands-on experience with these technologies. So that's the sort of philosophy, but in a way, the deeper thing, and the thing that the Howard Hughes Medical Institute sort of wanted to happen when they started this program, was they knew that investigators from different disciplines were happy to work together. But they also knew that it was hard because it took time and effort. It was frankly often time and effort they didn't have, right? Whereas smart students who were kind of motivated by this type of, you know, interdisciplinary science could be the people who would actually make this possible. So this was actually what the Howard Hughes Medical Institute told us in 2005 when they announced this program. And we became over 10 universities to get that original award that led to this program and the NIH funding. So in a way, that's the real thing here. So we have this multi-scale theme. It's nice. It's sort of this biomedical science neutral, allows us to cover a whole lot of different technologies and integrative approaches. It gives us a sort of common language in physics and measurement and stuff like that. But it's doesn't really exclude. There's not much that any one of us would do that would exclude. So what in a way makes this makes this more special and more important is the opportunities you get to talk to each other and learn from each other and work together in the labs. Okay. And you know, what I want to do is, it's amazing over the 15 years that this program's been running that we have somehow always been able to offer these labs and always been able to, you know, kind of keep this program going, even though no one's sort of paying for it. It's just the faculty want to do it and the students want to do it. So we want to have you part of it. But in a way, the real success is when you get together and do something new and exciting. So when one of you hears something from Kristen and says, you know, my advisor or my advisor's collaborator does X, and maybe we could look at this on, you know, sex differences in disease burden. So that's really what it's all about. So what I want to do is make sure these sessions are good for that. And I have to say already, this room doesn't thrill me because where we used to have it, we could sit around a table. We'll work on finding a better location. But also we don't have to just do this. You don't have to just talk to each other. Will and I were trying to look for the room. You know, he reminded me that one of his co-mentors is a new faculty member. We actually had a ton of super exciting new faculty members who have joined UCSD during the pandemic, who many of us are still just getting to meet for the first time. I met one of them from biology the other day, who would be such a great faculty member for this program, but you didn't even know about it. We did have one of those new faculty members, Dr. Fahad, give a talk at one of these last year. So we may want to use this opportunity to get some of these new faculty members who don't know us and who we don't know come to talk to us too. There are other things we can use this time for too. We can use it to talk about how do I do better interdisciplinary science? How do I manage having 2 mentors or 3? You know, Gabby's really like 5. She just you know, throws their hands up. How do I, you know, what happens when one mentor says, oh, let's write a paper on this, and the other one says, oh, let's write a paper on this. You know, how is this interdisciplinary training of mine going to help me get a job in academia if academia is still, you know, being traditional and wanting to know? And so anyway, I just want you know, this is basically your time, and I want you to use it. And one of the ways you should for sure use it is to share your own progress and stories. Just the opportunity in a small sort of friendly group to just get up and talk about your research is, you know, maybe you do it in lab meetings, and maybe some of you do it in meetings and other things, but for many of you, it'll be the first time you've done something like this, and just being able to do it regularly is really good for you. So I would definitely encourage you to keep doing that, but you know, you don't just do that. You can do lots of things. So, and think of this as your time, and tell me or Kelly when you have ideas, someone you want to hear from. Like we in the past, we had discussions about standards for data, how to do presentations. Tell us what you want, and we'll find people. We can get back the same people who gave us talks on how to give presentations. We also have some cool alums. You know, one of my personal favorites is Samantha Jones. She was a chemistry and biochemistry PhD student. And if you ever listen to the American Chemical Society podcast, which are really popular, you'll hear her. There's 2, one called Solutions and one called Orbitals. One of them she produces, and the other one she actually is the host of. So she was an interfaces student, and she has actually come back and talked to students here about communications. One of them, Britton Boris, was actually a co-author on the science paper that announced the discovery of Paxlovid by Pfizer. So he's got some interesting stories to tell. Some of them have gone on to faculty careers. and have, you know, great advice. So anyway, just tell us what you want to do at this time.
Thank you, Dr. McCullough, appreciate that. Just to let you know, we will be having a mixer next Thursday, so before you go out for Halloween, come on by. Don't know actually where it's gonna be at this point. Would love to hear suggestions from you about where you'd like to have it. And that's a great opportunity for you to just sit and talk with some of your friends about, what you're working on and about who you might like to collaborate with, and I'm always open for suggestions for people to speak to the group about different topics, you know, it can be about, you know, potential careers or presentations or, how to get funding for research later down the road, so just let us know, we're happy to try to make this as beneficial for you as we can. Shivani, I don't know would somebody tell me what time it is? I don't know how much time we have left for Shivani. 48. How fast can you talk, Shivani?
Very fast. I'm from the East
Okay,
Coast.
all right. Okay, everybody, this is Shivani Shukla, and she's gonna speak to us, and we may have to go a few minutes over.
Okay.
And we do have some snacks that you'll unfortunately have to get on the way out because of timing today, so I apologize for that. Again, this will get better as we go forward. Bianca, can you come help?
Really?
There you go.
And do I need to show a screen? Okay.
I'm sorry, Shivani, I have to go to that office hours,
Oh, yeah, no problem.
but thank you for volunteering.
You see it?
I don't know if you're sharing on those.
You'd like to share your entire screen, and not just the.
I can see it. It was good, yeah. I'm definitely screen sharing that says. No, I can see your screen. Okay. Yeah, we're good. Hi, everyone. Thanks for being here. I'm Shivani. I'm in the bioengineering department, but I work in nano engineering lab with Dr. Zainab Jahed. And I'll be talking about a nano electrode platform for electrophysiology of different cell types. So as you guys might know, different tissues in our body are electrical in nature. So neurons in the brain fire signals, as do heart cells and muscle cells, and the individual spikes that they fire are called action potentials. And so we can use a lot of different techniques to record this electrical activity, and as I'll talk about, it gives us a lot of important information. So this is just an overview of electrophysiology tools for recording from the brain. So you probably know about MRI. And within the same like spatiotemporal resolution, we have EEG, which is like the big electrodes on someone's skull, and that can help us detect like epilepsy, or measure sleep waves. And then at greater spatiotemporal resolution, we have electrophotography, Utah arrays, which is like a bed of nails that you put into the brain, and microelectrode arrays, and these record what are called local field potentials. So each electrode is measuring the electric potential or the electric field around the electrode, which can consist of many different cells firing at once. And then the highest level of spatiotemporal resolution is the patch clamp or the nanoelectrode array. And so these tools can go into single cells and record the actual action potential with the truest waveform like amplitude and full width half max that is possible today. And
there tends to be a trade-off between spatiotemporal resolution and clinical relevance, because you can imagine that like sticking an electrode into someone's brain into a single cell anywhere in the brain would probably disturb the brain. Switching back to the heart, I'm just going to review the cardiac action potential. So this is governed by four main ion channels: the calcium one, the sodium one, the potassium one, and the chloride one. And so you can see on the bottom right, there's a picture of the sarcomere, which is the basic unit of contraction in the muscle cell. And so to begin, when a cardiomyocyte receives a signal, all of the sodium ion channels open and sodium rushes into the cell. And so this depolarizes the membrane. If you remember, the plasma membrane has different concentrations of ions, all different types of ions across it, and that creates like an electrical potential. And so when sodium ions rush into the cell, that changes the potential. So it changes the voltage to a positive value. And then after that, the chloride and potassium ion channels open, and those two ions leak out of the cell. And so then the cell starts to reset itself. And after that, the calcium channels open. And you can see, if you remember from biology, the rush of calcium into the cell is what causes the sarcomeres to contract. And so at this point in the muscle, the entire muscle is contracting. And then finally, all the other channels have closed, and only potassium is leaking out of the cell to reset the membrane potential. And at this point, the muscle can relax again. So you can kind of see that this like highly orchestrated set of events gives the action potential a very specific shape, and it also enables the contraction of muscle. And this shape is actually really important because in preclinical studies, the FDA wants to see that any drug that you invent, if you change the dose of the drug, how does it affect the shape of the action potential? Because then that tells you how does this drug affect specific ion channels in the heart. And actually, different cell types in the heart have different action potential shapes. And the accumulation of all of those shapes gives you the ECG signal that we use clinically to diagnose cardiovascular disease and to treat them. So you can see that the action potential is a powerful thing to measure, but we can only measure it using intracellular recording. We can only measure it if we go like into the cell with an electrode. That's very invasive. It's ideally what we would want, but it's much worse clinically than extracellular recordings, which are non-invasive. They can be higher throughput, and they show like the big picture of what the actual organ is doing. And so in my research, I try to record intracellular signals and extracellular signals and compare the two and see if we can use computational models to predict the intracellular signal from the extracellular one. So I talked briefly about nanoelectrode arrays, which I said are like one of the intracellular recording tools. And a lot of labs actually at UCSD have developed nanoscale electrodes that could go into the cell and measure the active potential waveform. So this is just an overview of them. My lab uses nanopillars. A lab in electrical engineering uses nanowires. And then there's also field effect transistors in the nanoengineering department. So that's just an overview. They're all pretty much the same dimensions and the same size. They just need to be small enough to deform the plasma membrane and give us access to the cell interior. Now, if some of you are familiar with electrophysiology, you might have heard of patch clamp, and you might be wondering what's the comparison between patch clamp and nanoelectrode arrays. Well, anyone who has done patch clamp will probably tell you that it sucks. Like every 5 minutes, a cell dies. And you're trying to measure from a tissue, you can only measure one cell at once because you have to use a tool called a micromanipulator to move the electrode around and like slowly puncture the cell. And so the nanoelectrode array platform, like these, have all been developed in response to the difficulty of patch clamp. So in the Jahed lab, we go through a cycle of studying the cell nanopillar interface. Then we design nanoelectronic devices to record the action potentials from cells. And then we take this data and we try to redesign our devices to get better data and also improve on data analysis techniques. So to start, this is one of the experiments where we're trying to understand the interface between neuron-like cells and a bed of nanopillars. And if you look at this, you can see that there's differences in cell morphology, such as the area that the cell has when it's on the nanopillars versus when it's not, and are currently in the process of conducting analysis on the neurite length and the neurite width in this like PC, like differentiated PC12 cell type to see if there's differences in that area based off of the spacing and the dimensions of the nanopillars. So then, after we study the interface, we can construct devices where this is a microscopic image of a nanoelectrode array where on the electrodes there are nanopillars, and you can see cardiomyocytes beating on top. And cardiomyocytes are nice to use because when they beat, they fire action potentials. So initially we see something like this, where we see like these tiny little spikes. Those are the extracellular signals that are being recorded from the nanopillar outside the cell. But then when we send a small electrical pulse through the conductive nanopillars, we start to see the action potential. And this is the main mechanism of how our devices work. So you might be wondering, does the electroporation damage the cell? You might have heard of electroporation in the context of transfection. This stimulus is 1,000 times smaller, and actually the cell membrane repairs itself. In the case of our nanopillars, it's not actually puncturing through the cell membrane, it's creating like nano-sized pores, which we can tell with electron microscopy of the plasma membrane. And we can see that over the course of like a few hours, the membrane heals itself, and then we go back to extracellular recording. So this is kind of a big picture diagram of what my devices look like. So when I'm holding it, it looks like that. There's like a well glued on top, so you can put cells and media inside. And then if you zoom in, you can see all the electrodes. Right now there's 60 of them, and then on each electrode there's a set of 9 nanopillars, and we could completely change like the pattern, like the array shape, the electrode shape, the nanopillar dimensions. So it's very tunable, and this becomes important when we're trying to tune it to record from different cell types. So just briefly, I work in the clean room a lot, and I start with a glass wafer, and then to fabricate the nanopillars, we use what's called a chromium hard mask, where we just deposit these dots, and then we etch down on the wafer, and the dots create like a mask, which allows the nanopillars to form. And then we do another process of etching with HF. And then we sputter on a conductive material such as platinum or indium tin oxide, and then we insulate the rest of the wafer so that the only thing that the cells are in contact with is glass or the conductive tips of the nanopillars. So this is just some more fabrication details. The chromium dots have wings, and that creates like a shadow on top of the wafer. And that's why our nanopillars look like have a pyramid shape, and they're not just like going straight down. And this improves the robustness of the device, and as well as the interface between the cell membrane, which has to wrap like very neatly onto the nanopillar and the device. So once we get a bunch of recordings, we can record from different cell types. So on the left, you see a picture of biofilms growing on top of our device. And then in the middle, you see some cardiomyocyte action potentials. And then on the right, you see PC12 like adherent cells, which are differentiated with the nerve growth factor. And you can actually see like the heat map is showing the spiking times of the action potentials across the culture. And you can see kind of the most coherent one is the cardiomyocytes, because muscles beat like as one, muscle cells beat together in a synchronized fashion. So you can see that a lot of those action potentials happen at the same time. Whereas in the PC12 culture, which act like neurons, such as like a neuronal network, the firing is happening in like localized locations. And then the biofilm one, we're still trying to figure out. And then finally, after we kind of get the heat maps and we get more data, we can redesign the devices to try to record intracellular and extracellular signals at the same time. And we can use this data to create computational methods, such as using machine learning to predict what the intracellular action potential would look like given an extracellular spike. So in conclusion, our lab has been able to study the cell nanopillar interface and create devices which help us record from 2D as well as 3D cell cultures, and then redesign our devices to be able to tell the difference between intracellular and extracellular recordings and measure the relationship between them. Thank you for listening. Let me know if you have any questions.
I had a quick question. You said that the FDA tests drugs to see if it has the current. So that's like all drugs or just drugs that have to do with like heart or neurons?
It really depends on like how the drug is being administered. Most drugs require this kind of testing because most of the drug gets to the heart anyway. For like drugs related to the brain, they do require a dose response curve, but they could use like CT scans or MRI to show that activity, and they don't need the actual action potential for that.
And how do people test it?
Yeah.
Did everybody already start clap?
Oh yeah. They got it.
So how do they test it that again? How do they get that curve? Where they-
The drug response.
Yeah,
So they do patch clamping right now. As they in like a cells in a dish, as they increase the dose of the drug, like the concentration of the drug, and then they measure the full width half max of the action potential, and then they plot that.
Anybody else? Did everybody get lanyards and pens and a bag up at the front? We got goodies. We also have, thank you very much, Bonnie, appreciate it. Appreciate both of you speaking today. We've got sandwiches back here, little half sandwiches. Sorry it was late. And, like I said, please send me suggestions. I'm happy to, move the location. The time is kind of fixed. We work around the trainee schedules for the time. So some trainees can meet on Tuesday from 11 to noon and some on Wednesday from 3 to 4. So I'm gonna try to alternate that, the time, so nobody misses too much. But, if anybody has any questions or anything, let me know.
Well, I'm gonna take that time.
Otherwise, help yourself to sandwiches. Are you Abby? I'm Kelly. Nice to meet you. We got sandwiches, well, vegetarian, so.
Oh yeah. We're using machine learning to, like, we have action potential data, and then we use ML to predict, that data. And keep it very on. So we have like, from the same cells, we record extracellular and the data, I mean, that is not done. Oh, that's what it's like. Really straight. Like, we put the cell on the device, we get the extracellular by, yeah, that is. Yeah.
Because I didn't know that.
Oh, yeah, like we input to the, which is not really, we input extracellular spike. And then we can like train it with the output, saying this action potential is the output. So like, how do you predict that? We ask the algorithm. To predict what the extracellular one will look like, given this extracellular.
I saw that.
Yeah, so there's a lot of physics that people use to try to explain. We essentially believe that extracellular spike is like the first derivative of the extracellular one, and there's like a circuit model that we use to predict what that spike would look like.
You definitely having some equivalent something for that.
Yeah.
That's a very, that would be
Exactly. So yeah, so that circuit model is still under development. And we have some idea, but we still get a lot of spikes. It's like a hybrid signal where the electrons like half in or out, and then we see a spike, and that's something that the machine algorithm can't tell. It'll be like, what's that? So there hasn't really been a comparison between what the algorithm puts out versus the circuit model and what it predicts from the circuit model using the actually. So right now the algorithm is basically a black box. It works pretty well, but it's, I mean, it's being developed by someone very well. Like, it's, they're making progress. It's doing well. Eventually I'll be working on that for neurons, since we're only doing like cardiac medicines right now. So that'll be like my future. Like, you got a crack shell? Me too. No, congratulations. Oh, yeah? I don't know. Okay, nice. I didn't want to do science. I think you think this was so we just wanted to know more about this. Oh, yeah, yeah, but yeah, if you wanna talk to her, yeah, it's a cool project. I haven't really started the ML part, so it's supposed to be that. They were gonna try one. Let me just take the goodies. Oh, yeah.
Thank you.
I mean, like. for the
At this
purposes of-
scale, something like. I saw, a while ago, I saw. took class with Shukla.
Or do we have any extra tumblers? Underneath the table, they're in boxes under there.
Scale